R. Alexander Pyron is the Robert F. Griggs Assistant Professor of Biology at the George Washington University. His research focuses on the deep evolutionary history and recent ecological interactions of reptiles and amphibians around the globe. Professor Pyron maintains active field work in Brazil and Sri Lanka. He received his Bachelor of Science in Biology from Piedmont College and MS and Master of Philosophy and PhD from the City University of New York. Before joining the faculty at GWU, he was a National Science Foundation postdoctoral research fellow in bioinformatics at Stony Brook University, New York. And with that, join me in welcoming Professor Pyron to deliver his lecture on the origin of amphibian biodiversity.
And our clicker. So what I want to talk to you today about is some of the research that I've been doing since I got to GW, trying to uncover the patterns that lead to, not only global variation, but the origins of biodiversity globally, and how that biodiversity varies through space. So the diversity of life, the number of species that we can observe just by looking at ecosystems either locally or in pictures from sites around the world, is really massive and it's really extraordinary. And it has a temporal component too. We can think of dinosaurs and other extinct animals that contribute to historical faunas, as well as the extant animals that contribute to the faunas that we see today. Things from birds to tuataras to zebras and frogs. And actually, looking at this, the only one of these animals I've ever actually seen is a peacock in the wild in Sri Lanka. But what we want to know is, where did it come from? How does it persist? And why does it persist in the configuration that it does? And what do I mean by configuration? And so, again, it's probably familiar to almost everybody that biodiversity varies globally in terms of where it's found. This is a picture from Antarctica. And when I found this picture, the main thing I was surprised was how sophisticated our installations are in Antarctica. I had no idea they had basically whole cities and stuff built down there. I thought it was just like igloos and things. But apparently we have pretty sophisticated installations. And yet there's hardly any species down there. The largest terrestrial species, I believe, are some invertebrates. Penguins don't really count because they're aquatic birds. So, there's hardly any land animals that occur in Antarctica. And yet if we go several thousand kilometers to the north from the Antarctic field stations, we can end up in the Amazon rainforest, where probably every single plant stem that you can see in this picture is probably a different species. That may be a slight exaggeration, but it's not too far off. And even though we can see forests that may look this green and this, lush, say if we went down to Florida or even if we went out, to the Appalachians, there most of the plants would be the same species, whereas when we get to the Amazon, everything's a different species. So we have this spatial variation in biodiversity that in one way or in one sense has a pretty obvious explanation. We intuitively know why there aren't species in Antarctica, and we intuitively know why there are lots of species in the Amazon. But what we really want to drill down to are the actual mechanisms that are responsible for those species being there and how they're maintained through time. And so one of the main patterns that we see going from Antarctica to the Amazon is something called a latitudinal gradient in diversity. And so this is just a picture of the density of mammal species where they're found throughout the globe from a study that came out a few years ago. And obviously the highest density of mammal species is in, say, tropical South America, tropical Africa, and tropical Southeast Asia. And then once you move towards the poles, you rapidly lose species. These lower two graphs are just quartiles of the rarest species and the most common species. So if we just look up here, we can see this latitudinal gradient. And latitudinal gradients in diversity are a very old and very well-recognized problem. They go back at least to, they were written out about explicitly by Darwin and Wallace, but they were noticed by explorers during the classical period and even back into the times of the Greeks. And what you see is a latitudinal gradient, an increase towards the equator, in species, which is the most obvious, but also in terms of things like communities. So maybe we go outside and we find 50 species of plant in the yard here, and that's our community. We could go to the Amazon where there are 10 times as many species, but maybe they all have very small ranges and they never overlap, and maybe at any one site in the Amazon we would only see 50 species, and the community diversity wouldn't vary. But that's not the case. Communities also vary as well. They get richer. We could find 500 species or 1,000 species at one site in the Amazon. And the maximum you would find here is, say, 50 species. Also, the traits that species exhibit. So Maybe we'll think about birds. Most of the birds around here just, eat seeds and stuff you can leave out on the bird feeder. Maybe you have woodpeckers that hammer into trunks or something. If we go down to the Amazon, also the traits that species exhibit. So maybe we'll think about birds. Most of the birds around here just, eat seeds and stuff you can leave out on the bird feeder. Maybe you have woodpeckers that hammer into trunks or something. If we go down to the Amazon, we'll see birds eating fruit, we'll see birds eating nuts. So, the amount of actual different biological pathways and different food webs and things like that that you actually get as a result of the interaction of these species in their communities with their different traits. I'm not going to focus so much on the latter two. Traits and functions are things that I'm starting to get into with my research, but what I really want to talk about are the two things that we can observe at the broadest scale, which is the diversity of species and the diversity of those species within communities. And as you can well imagine, this is not poorly trodden ground. If we start to look at historical hypotheses for what resulted in these patterns, you have things like the mid-domain effect, which just suggests that if we actually took species ranges and just shuffled them randomly in space, we might actually expect to see an overlap in the center of their range, just as an artifact of complete random overlap. You could have something like maybe geographic area. Maybe the tropics offer a larger area to species. Or maybe there's a species energy relationship. Maybe the higher energy in the tropics contributes to there being more species. Or maybe a whole lot of things. So maybe climate harshness, stability, historical perturbation, evolutionary rate, time, speed, biotic interaction. The list goes on. And there's lots and lots of papers that review these mechanisms as well. And if it seems like a random hodgepodge, I think it's because it is. So what we really want to do, and one of the other things that I kind of want to make a point about, is sort of how we approach complex problems like this in view of the scientific method and how we can try to simplify the mechanisms that might be responsible for observed patterns that, if we don't focus on the underlying causes, might result in sort of a panoply of confusing and potentially interacting or potentially mutually exclusive hypotheses. Because not all of these are mutually exclusive. Maybe 4 out of 11 of them could actually be true in any one group. But the question is, do they satisfy the fundamental mechanisms that are responsible for species arising and species being maintained through time? So what are those mechanisms? This is some data from some work I've been doing on frogs. It's actually a separate paper, but it's still amphibians, where we can look at frog diversity on islands. And if you've done any reading in ecology and evolution, you've probably heard of island biogeography theory developed by E.O. Wilson and Robert MacArthur, which suggests that as islands get larger, they'll have more species because there's a larger area for those species to colonize. And that appears to be partially true, except we've got all these dots along the abscissa, islands with no species. And those are islands like Greenland or islands off the coast of Antarctica. And they're huge, but they don't have any frogs because there's no continental species pool of frogs to sample from. And so, okay, on one hand, you might say, well, that's kind of obvious. That doesn't really tell us anything about why there are more frogs on this large island, which is probably New Guinea, than Madagascar, than Borneo, than Sri Lanka, or something like that, as they decrease in size. And when I actually first got a grant to work on this, I was trying to explain it to my uncle, and he was like, well, what's a latitudinal gradient? I'm like, well, I'm studying why there's more species in the Amazon than there are in Antarctica, and he just kind of looked at me. And so then we could say maybe it has to do with productivity or the amount of ecosystem energy, which is another major facet, sort of an obvious conclusion that you might draw just if you only knew a little bit about the system. And again, that has a strong relationship. It's not massive, so islands with more productivity seem to have more species, but then you've still got these islands here that seem to have pretty high productivity that don't have many species on them. So let's zoom in a little bit. What's our global set of islands? Let's isolate it a little bit more and just look at islands that are around, say, 10,000 kilometers square. If we look at these islands, there's a pretty massive variation in the number of species that islands have when they're that size. And the same for the primary productivity of those same islands. Pretty big range in productivity, pretty big range in the number of species. And what's this little dot at 0 in both of those graphs? A large island with high productivity. It's the big island of Hawaii. So it's obvious why Greenland doesn't have species. It's cold, there's nothing there. But Hawaii is a tropical paradise. It has hundreds of species of birds and flowers and all sorts of things that would make it great for frogs. And the reason is that they just never got there. If they got there, if we shifted Hawaii over and put it next to Cuba, it would probably have hundreds of species of frogs. And in that isolated case, it's kind of easy to say, well, okay, well, there's no frogs there because they just didn't get there. But the point I want to make about all three of these things put together is kind of a fundamental point about the mechanisms that actually generate biodiversity and are responsible for its variation through space. Because only three mechanisms can actually change the number of species that exist and change the number of species that are in an area. And those are speciation, the accumulation of new species as they divide through time, which is sort of the be-all, end-all. If we didn't have speciation, we wouldn't have species and there wouldn't be latitudinal gradients. There's also extinction. We can lose species differentially. Maybe we're losing more species on smaller islands even though they're speciating at the same rate. Or maybe extinction is tied to something like the amount of energy in an ecosystem. And then there's also the historical process of dispersal. The frogs actually have to get from place to place. So maybe speciation gives us 100,000 species of plant in the Amazon, but if they never jump across the Atlantic to, say, sub-Saharan Africa, there won't be any species there. And on the one hand, it's easy to look at that and say it's kind of a truism, that those are really obvious processes that affect biodiversity, but that it doesn't really tell us anything about why species are where they are. But I think if we extend those through time and think about how they actually lead to diverse assemblages, that'll actually become more important and also allow us to do the third thing that I kind of want to talk about, which is linking the deep evolutionary history. So the things like continental breakup have occurred over tens or hundreds of millions of years with ecology, because the frogs that are here in the DC area are reacting to rainfall on a year-to-year basis. So we want to link across timescales and also integrate these three processes or these three mechanisms that actually generate biodiversity. And so I think the easiest way to visualize this is if we back up and take the planet as we have it. We'll pretend that plate tectonics doesn't happen and that all continents have been where they are forever, just because it's simpler that way. But imagine a world where there are no species. How do we then, from that point, generate the observed patterns? So at some point, there was only one amphibian species, and one tree species, and one bird species. Everything condenses back to a single common ancestor at some point. So what we can do is look at this globe and say at one point there was a single amphibian species, and it existed somewhere here, and then through time has dispersed from continent to continent, and also undergone speciation in those areas, as well as extinction in those areas, and that's what's led to our biodiversity gradients and general patterns of biodiversity. So, and that's what really, I think, highlights the importance of considering these three factors independently, speciation, extinction, and dispersal, because we could say maybe amphibians originated in South America, maybe that first species was there, and then it subsequently dispersed, which means it's been here for a long, long time and is going to accumulate a lot of species. Or maybe it was somewhere in the tropics, but also disperses really quickly. So amphibians can just hop around from continent to continent. And that's why you have a rapid buildup of speciation. Or maybe speciation is highest here, so it doesn't really matter. So the ancestral amphibian was tropical, but you have higher speciation here and higher extinction in the temperate zones. If that were true, that would immediately lead to a latitudinal diversity gradient. Spoiler alert, it's all of those things, but we'll get to that later. But if we reduce it down to just those core factors, we can kind of cut through more than 100 different hypotheses that have been proposed and try to generate some sort of generalized model that accommodates the three and the only three factors that actually will lead to spatial and temporal variation in species richness. And what we need to do that is something called a phylogeny. And so if a phylogeny is just a diagram that shows the evolutionary relationships between species. So if we have all the species of amphibians sampled, we need to know how they're related, because that'll tell us if the amphibians in South America are related to the ones in Africa, or the ones in Australia, or the ones in Europe, and allow us to trace these processes through time. And we can also try to estimate extinction from phylogenies as well, because we can make a phylogeny of taxa that we see today, but then there's also all these other branches that have gone extinct that we can't really put into a phylogeny. But sparing you the math, we can try to use the information from the phylogeny to try to estimate how much extinction has gone on and to see if that's related to other things like climate or energy. And so there are existing frameworks for the molecular evolutionary timescale of most species. You may have seen this diagram. the time tree of life. And so what we can do is take molecular genetic data, or DNA sequence data, build phylogenies, and convert them to time trees, which gives us a temporal scale as well as an evolutionary scale, or a relatedness scale, with which we can compare amphibians, or any group from around the world, as well as communities, and I'll get back to that later. And again, a large part of my research has to do with paleontology of amphibians and fossils and how we actually calibrate how old this tree is based on what we know about the fossil record. I'm going to omit a lot of that just for simplicity. So just bear with me when I say that I have a dated tree and just assume it's true. It's a lot easier that way. And it'd be a lot easier if the reviewers thought that too, but whatever. So, and this is as close as I'll get to bombarding you with math, but if we have a phylogeny, you know, we have these branches that connect the extant species, and then there's these ones we don't see that are extinct, and those get broken out, and so it leaves us with a collection of branches that are calibrated to time, say in millions of years here, that are related to the phylogenetic structure. And even if we take away how they're related, we just have this collection of branches through time. All other things being equal, longer branches mean lower speciation rates, because the branch keeps going and keeps going and keeps going without splitting in two. Shorter branches mean faster speciation rates. Extinction has an effect too. You know, a really long branch is more likely to mean low speciation than it is high extinction, because if there were high extinction, the branch probably would have gone extinct. So just in general, we can then try to ask whether or not the length of branches is related to, say, traits. Maybe these long branches are larger body sizes and these shorter branches are smaller body sizes. And so maybe smaller body sizes diversify faster. Maybe they're areas. Maybe these long, low speciation branches are temperate animals, and the short ones are tropical. And then the last thing that will link this back to communities is maybe it has to do with diversity. Maybe the more species we generate, the harder it is to make new ones. Maybe communities can actually begin to fill up with species that actually begin to saturate the available niches. And that may actually exert an influence on not only speciation rate, but also the diversity that you see within areas and among regions. So we could imagine perhaps that the tropical rainforests simply have more open niches that species can move into, whereas maybe temperate environments have fewer, and we'll come back to that later. So what we want to do is apply this to a group that's interesting, and of course amphibians come to mind. They're almost as interesting as snakes, which is what I normally work on, but these will suffice for the time being. And so we have frogs, we have salamanders, and we have a group that most people aren't familiar with called caecilians. There we go. So caecilians are these small earthworm-looking amphibians. There's only about 150 of them. Most people haven't heard of them. They occur in South America, Africa, and parts of Southeast Asia. They're kind of a relict group. I don't like the term living fossil, but it's sort of that idea. They're very old and very, well, they're not that boring. I was really excited when I found that one, but just generally they're kind of boring compared to frogs. And there's about 7,000 species of amphibian, one of the more diverse groups of terrestrial vertebrates. And the distribution of this species richness is really astounding. So one of the main places I do field work is Sri Lanka. And these are all frogs from a single genus, Pseudophylautus, the bush frogs. They occur, you know, in bushes, somewhat unsurprisingly. And on a single night hike in Sri Lanka, we can find up to 20 or 30 different species of these, just walking along, shining with our headlamps, until you see them light up because they kind of glow in the trees. And all of these have ranges of, say, a few hectares or a few square kilometers in these small, isolated pockets of rainforest in southwestern Sri Lanka. And so this generates hundreds and hundreds of species in this island, and yet they're able to persist through time with these massively small ranges in this massively small area. Whereas in contrast, there's a newt in Siberia that covers almost the entire Asian continent, and it's one single species. They've looked at it, and it's the same, you know, in the Kamchatka Peninsula as it is on the, you know, Finnish border or something. Across that entire span, no differences. And yet all of these frogs can be found in a single night in Sri Lanka. And those are just the Pseudophylautus. There's plenty of other frogs. You've got like the big long-limbed foam nesters and the meat-sized tree frogs and the toads, and there's tons of frogs there. I'm hoping to go back in about 2 weeks. And so this is what we see in the tropics compared to somewhere like the temperate zones where there's one newt in all of Russia. I think there's an in Soviet Russia joke in here somewhere. I just don't know what it is. And so if we look at this on a map, we can see this, again, latitudinal gradient. Looks a lot like the mammals. Looks like the same pattern we've already seen. This is our known diversity. Obviously, there's some holes. I don't believe that the central Congo rainforest only has as many species as Great Britain. I haven't been there, but I suspect it has more. So a lot of this is sampling bias. But what we see is that in tropical South America, there's about 2,500 species of amphibian. And the gradient is not only latitudinal, it's also longitudinal. There's only around 800 species in tropical Africa and 800 in tropical Southeast Asia compared to 2,500 in South America. And there's also longitudinal gradients on the continents. If we look at the eastern US here, if we go an hour and a half west into West Virginia and hit the Appalachian Mountains, we can start finding hundreds of species of salamander. It's a huge hotspot for salamanders. There's also another hotspot in Central America, but even in the temperate zone in the US, in the Appalachians, where it gets cold, snows, you wouldn't think it'd be real hospitable. There's tons of species. Same thing happens in China, so temperate eastern China, Japan on the eastern side of the continent. And you say, well, it has to do with the movement of the prevailing currents and warm, wet air masses. And so obviously the correlation here is strongly between warm, wet areas like tropical rainforests or eastern side of continents and having continuous currents bringing in water and dumping it on the land, and you've got amphibians there. Except they're not sea monkeys. They don't pop up when it starts raining. They had to get there. So what's our historical framework here? Does maybe having higher temperatures causes higher speciation rates? And so maybe the original amphibian was here and has diversified in the mountains. Or maybe it actually causes higher dispersal. Maybe all the amphibians are being generated in South America, but they know that it's nice to summer in the eastern US, and so they come up here and they establish there. But no matter what, these ecological correlations that we see, they're very strong, suggesting that amphibians are related to these warm, wet habitats, has to be explained historically by some factor of speciation, extinction, or dispersal. And so what our primary questions are is, where did amphibians originate geographically? If they originated in tropical South America, that would be pretty convenient because that's where most of them are, and we could say, well, they've been there the longest, and so that's where most species are. Maybe they only got to Russia recently, and so there hasn't been enough time for speciation. Or conversely, what if they actually originated in the northern latitudes, say in the temperate zones, say in North America or in Europe? That means that they have been there longer, and there must be differences in speciation and extinction. So which regions have been occupied the longest? And then do rates vary? Does speciation actually differ between, say, Western Europe and tropical South America? Or does extinction differ? And if it does it vary predictably in response to something like climate, which seems pretty obvious when we look at the map, but we still need to test that? Or does it vary by region? Can we say that species don't vary differently by rainfall and temperature, but maybe they vary, maybe speciation is much higher in South America? And does it vary with diversity? Can we look at the species that occur in communities and see if that's also related to the rate of speciation or extinction, which would suggest that there's some kind of ecological community level feedback that's influencing the ability of species to coexist locally? Because again, it's sort of what I alluded to earlier, kind of a tension, an intellectual tension, where we know this is occurring over millions or tens of millions or hundreds of millions of years. And yet when we go out into an ecosystem or an environment, what we're seeing is the day-to-day interactions of species and what allows them to interact, to coexist, to not compete with each other, or to not be outcompeted for resources. And so any explanation is going to have to integrate those things. So what we need now is a very large phylogeny. Conveniently, I built one. So this has about 50% of the world's extant amphibians that we were able to obtain DNA sequence data for. And again, I'll just gloss over how we actually build these trees. It doesn't really matter. But so we have around 2,400 frogs, which is about 35% of frogs. We have most salamanders. There aren't nearly as many of those as there are frogs. So we got most of them sampled. And about 41 caecilians. And we had a good sampling throughout the genome, so we can use the molecular sequence data to build this phylogeny. And so we can estimate from these little branches on the tips whether or not, well, first we can say where do they occur and then try to reconstruct their ancestral habitat. And then we can also ask whether rates of speciation and extinction vary. Conveniently, another group with the IUCN had generated range maps for every single species of amphibian. So I was able to just download those, overlay those on the globe, and extract for every species some measure of its ecological preferences. So if you're not aware, there's geographers have done an amazing job of giving us high-resolution files that say what's the temperature at every site on the globe, precipitation, radiation, things like that, amount of productivity from the plant mass. So I was able to basically get all of that and quantify the ecological niche of all of these species to then map on the phylogeny. And so the first thing we did was we looked at some tree-based analyses, so asking, using two algorithms, Quasi and Jesse, whether or not climate, so the temperature, precipitation, energy, or the area, whether or not they're temperate or tropical, causes rates of speciation and extinction to vary. Somewhat unsurprisingly, it does. So this is our same map here. This is just a plot of the species richness by latitude. These gray bands, if you remember the very first thing I mentioned, random range shuffling, those gray bands represent that. So clearly we can see there's way more species at the equator than there should be and way fewer towards the poles. So about 1,000 species at the equator, dropping down to nothing past around 45 degrees. And then what this big tangle of branches is showing us is this axis here is the ecological niche. Higher values are more temperate, lower values are more tropical. And most of the branches are here where the niches are more tropical, which we already knew that because we can see them on the map. But if we plot extinction and speciation rate, imagine turning this figure counterclockwise 90 degrees. These are the temperate species with high extinction, and when they move into the tropics, extinction drops off to almost nothing. Conversely, speciation starts out relatively low, but then when you get into the tropics, increases drastically. And this dotted line shows the difference between them, the net diversification rate, the net difference between the species gained and lost, which jumps drastically as well once you get into a tropical niche. And this is just a plot of latitude with the ecological niches of every species. So most species you can see are between 20 degrees, where they have a very low tropical ecological niche, very high speciation rate, very low extinction rate. It's a pretty straightforward explanation for why we see a latitudinal gradient. Tropical regions have much higher rates of speciation and much lower rates of extinction. This is corroborated by the analyses looking at area. I don't have a good figure for that, but it just basically says that if we classify species as temperate or tropical, just based on where they occur geographically, then tropical species have much higher rates of speciation, much lower rates of extinction, and vice versa. It also tells us that the rates of dispersal from the tropics to temperate regions are very low. Species want to stay where the habitat is good and they never disperse into tropical regions, whereas the converse is also true. Species in less hospitable temperate habitats frequently will disperse into the tropics, so entire lineages will move. And you can imagine that happening maybe through the Isthmus of Panama as they make their way down through Central America, or somewhere like Africa or Asia, it's even easier since it's a continuous landmass. So you have a lot of movement of species from north to south and very little movement from south to north, or from tropical to temperate. It doesn't answer all of our questions, though. One thing we might want to know is, if we break this up into groups that are ecologically distinct, like say tree frogs or toads or something, do we see the same patterns in all groups? And is there really a latitudinal gradient in things like the climatic variables that actually seem to be influencing rates of speciation and extinction? So the next thing we can do is break up the tree into what are called clades. A clade is just a group of related species. In this case, we have 66 families of amphibians. This includes like the toads are a family, the tree frogs are a family, salamanders form a bunch of different families, and we have 66 of them. Then we can look for latitudinal gradients in things like the rates of speciation and extinction, just to make sure our clade-based analysis corroborates our tree-based analysis. We can estimate the ages of those clades, because older clades should theoretically have more species because they've been around longer. And we can also see if things like area or energy of the clades themselves vary latitudinally. And I apologize for the cardinal sin of a large slide full of numbers, but what I want you to see here is that species richness, well, these are all significant. So there's a massive latitudinal gradient species richness. Families become more species rich towards the equator. If we jump down here, speciation rate gets higher towards the equator, and extinction rate gets higher towards the poles. And then the area and energy of clades also shows a negative latitudinal gradient. What's interesting and what can get us back to the community idea here are two things. One is that the ages of the clade, and we'll say what this means in a second here, are significant, as well as the estimated carrying capacity. So we can actually use mathematical models to estimate how many species can coexist in a community from the phylogeny. And communities that are farther north have lower carrying capacities based on our data. Saturation is just the observed number of species divided by the total carrying capacity. Clades that are farther north are more saturated. Clades that are farther north can have fewer species overall and are closer to that limit. And we'll come back to that idea in a little while. The age thing, this is kind of an aside, and it's kind of a subtle problem, but it's really an interesting thing to look at. On a phylogeny, say we have two species in family A, we can estimate how old those two species are when they diverged, called their crown group age. And we can estimate their stem group age, which is when they diverged from their next closest relative. So the crown group age is always younger than the stem group age. But what's interesting is that there's a massive difference here. Species richness is related to the crown group age positively. Older clades have more species, just what we'd expect if they're speciating over time, for their crown group. Species that have an older stem group age that diverged longer ago have fewer species, which is really counterintuitive. So what this is saying is that if two species diverged, say, 1 million years ago, but they diverged from their closest relative 100 million years ago, they have fewer species. Whereas a clade that diverged from its closest relative, say, 20 million years ago, has more species. It also varies latitudinally. Stem group ages are older at higher latitudes, which is telling us that the oldest amphibians are at higher latitudes, the oldest groups. They've been in temperate regions longer, and the crown group ages are younger there. And the only mechanism that I've been able to come up with to explain that is if there is high extinction in temperate zones. Because what that means is that even though the clades are sticking around at higher latitudes, you have more extinction, which is killing off any species that arises, which means you're going to have a younger crown group because the only species you see are going to be relatively recently evolved because any older species will have been trimmed off by extinction. Whereas in the tropics, that action of extinction is not nearly as high. And so you have much older clades that aren't subject to long-term extinction pressures that thus still have more species. This is a plot of going back to the carrying capacity. So from the phylogenetic analyses, we can estimate how many species could theoretically occur in a clade if there's the signal of species richness driving down speciation rate. And there seems to be in all of these clades. The more species that you have, the longer the branches on those species get, which means that more species are slowing down the rate at which you accumulate new ones. The ecological theory behind this is that if these are all competing for niches in environments, then as you generate more species that are occupying more niches, there's going to be less available space for new species to diversify into. And so as you have less niche space available for new species, the rate at which you accumulate them is going to decrease. And that shows a strong latitudinal gradient. So at higher temperate latitudes, you simply can't have as many species. And there's not been a real explicit test of this, but what we see is that, at least from kind of superficial readings of the literature, when you do coexistence experiments, it seems like, at least in amphibians, that the species from tropical regions have more specialized niche requirements, which means they're more finely subdividing the available ecological space, which would, given the same or even a larger total niche volume, allow more species to coexist locally in the tropics before that feedback on speciation or extinction occurs. And that fewer species can occur at high latitudes, and as a result of that, you're going to have much higher extinction pressures, which is exactly what we recover from the clay-based and the tree-based analyses. There's still one more thing that we want to look at, though, which has to do with area and time of occupancy. So we said that, say, tropical South America has 2,500 species, whereas Africa only has 800, or Southeast Asia only has 800. What we want to know is if that's related to the length of time it's been occupied, because if South America has been occupied longer than Africa or Southeast Asia, it makes perfect sense. So what we can do is divide the globe up into a set of different ecoregions, and these are ecological geographic areas that are separated by having different land masses or different faunas on those land masses. And it's pretty straightforward, things like tropical South America, Middle America, North America, Western Europe, Eastern Asia, the Palearctic, Australia, Sub-Saharan Africa, that sort of thing. And so we can try to find out if there's differences in the length of occupancy of these different regions. So we want to know if time varies between them. We can also estimate speciation rates and see if those are related to the area of those regions. Maybe larger areas have more niches open and cause species to accumulate more quickly. So we can try to relate that to time and rate. And what we actually find, interestingly enough, is that there's no effect of time. Time has no effect on species richness, whereas the diversification rate in areas is strongly related to the species richness, and the diversification rate in areas is strongly influenced by the energy in those areas. Higher energy areas, like the Amazon basin, have much higher throughput of evapotranspiration of water, and that appears to be strongly related to an increase in the speciation rates, which feeds back into an increase in species richness. Interestingly enough, and what I alluded to earlier, temperate regions have been occupied by amphibians for much longer than tropical regions. So all of the fossils are in temperate regions, going back hundreds of millions of years. And also all of the most ancient amphibians that we see alive today are in temperate regions. In the southeastern US, we can find 150 million-year-old amphibian families that have, we don't necessarily know where they persisted, but there are all of these temperate lineages, and the same is true in Western Europe, all these very, very ancient frogs. Tropical South America is the second most recently colonized area. The only one that's been colonized more recently was the West Indies. But tropical South America was only colonized by extant amphibians around 90 million years ago, whereas amphibians have been present in temperate regions in North America and in the Palearctic in Europe for well over 200 million years. So that action of extinction that's changing those crown group and stem group ages has been going on for a long time. And only very recently have these tropical regions actually been colonized by these lineages. And then what we've seen happen is that because the speciation rates are much, much higher there, they're just rapidly exploding in diversity. So the latitudinal gradient in amphibians seems to primarily be unrelated to the time of occupancy. It doesn't matter how long the amphibians have been somewhere because there's such a strong effect of speciation and extinction, where the richest areas are actually very young, and you have ancient clades with hardly any species in them. And those rates vary latitudinally, which is not unpredictable. It's not an earth-shattering conclusion. But what's interesting to see is that it's a difference in not only speciation rates, which are higher in the tropics, but extinction rates, which are lower in the tropics and higher in temperate regions. And so we have variation in both of those, and also that imbalance in dispersal. Species are pretty keen to disperse from temperate regions into the tropics and diversify heavily there, whereas they very rarely disperse out of the tropics. And so we see a strong impact of climate. But what we want to know is how. So for the last part of the talk, I'm going to kind of sort of speculate a little bit, perhaps wildly, about why these climatic variables or these ecological variables have such an impact. And going back to the very first slide where I had all those potential explanations for why you had latitudinal diversity gradients, one of them is the idea that maybe higher energy environments cause molecular evolution to happen more quickly. But that doesn't actually seem to be the case. The rates of molecular evolution, just the changes in the DNA and the sequences in the genome, seem to occur at the same rate. So then how then can we link these ecological factors like temperature and precipitation to patterns of diversity and to different rates of speciation and extinction? And the carrying capacity analyses that I've mentioned, I think, provide one at least fairly cogent possible mechanism for that action, even though we need a lot more additional testing to actually establish a firm link. But going back to the idea of hiking around in the tropics and finding frogs, if we go to, say, northeastern Brazil, which is the other place I do field work, and I'll hopefully be going back in June, we can walk along and find all sorts of different frogs, just in one area, in one small region, in one night. And they occupy very different ecological spaces in the environment. This is a toad, and it has much thicker skin and survives in drier parts. Then you have puddle frogs that occur, in small temporary puddles that arise during rainstorms. Then you have massive toads that are the size of beach balls that occur deep in the forest and can eat mice and birds and things. And then you have crazy tree frogs with these, spatulate noses and things that occur on only certain size streams on certain size trees. And then in the swamps, you have smaller tree frogs that can breed there. And there's 4 or 5 different species of small tree frog that can all breed in the standing water that accumulates, at lower elevations, closer to the water table. Even in those regions, you have ecologically differentiated species like sheep frogs. They're larger prey. And then up in the branches of the trees around those standing areas of water, you have the medium-sized and the larger tree frogs. Then you have a whole other ecological guild of the smoky frogs or the rain frogs. These are large terrestrial frogs that cover vast amounts of distances during the day. They're basically hopping around anywhere you are, and they're very opportunistic. They eat everything. Some of them get massive, and they secrete these toxins where you can't really touch them or your face will swell up. And then you have smaller sheep frogs. It keeps going. Some are fully aquatic. These are all from one area, one pond, basically. And then you have sort of intermediate toad-like hybrids. These are all occupying different ecological spaces in the environment. And they're all finally partitioned or subdivided the available niche space. So I've kind of touched on the main points already, which is the idea that, at least from an ecological standpoint, what appears to be happening is that in tropical environments that have higher, say, energy throughput and have broader, especially in montane regions, you have a very, very broad range of temperature and precipitation and ecosystem function. But it's also fairly stable and can be very easily broken down into small pieces. So the temperature doesn't really change that much, say, at a single elevation on a mountain. And so you can break that mountain up into a bunch of different layers of different temperatures, if temperature is what is important. Whereas in a temperate region like the US, a single point on a mountain will experience the entire range of temperatures throughout the entire year because it varies so drastically. So in the summer it'll be hot, in the winter it'll be cold. If we go back to this site in Brazil, in the summer it'll be hot, in the winter it'll be hot, and I promise you it's very, very hot. But what seems to be happening when we combine that ecological data with the phylogenetic data and the signal from the branch lengths that tell us about rates of speciation and extinction is that there appears to be a broader amount of open niche space, of open ecological opportunity, we can call it, in tropical regions. And that's caused by the increased precipitation and temperature and energy. So the effect of climate seems to be to make more niches open in tropical regions, which can then be occupied by more species, which can diversify at a faster rate and don't saturate as quickly. Whereas in temperate regions, there appears to be a much smaller range of available niches that can only be occupied by a much smaller number of species. And those species also have to be far more generalist in their approach to the general ecological utilization of resources to survive those massive temperature swings. And the signal that we get from the phylogeny is that groups that are closer, or that are farther away from the equator, rather, are also closer to their saturation and probably have been for a long time. So not only can we have fewer species here, we have about as many as we can have in temperate latitudes. Whereas in tropical latitudes, there's large open niche volumes that can support much more diverse communities before you have ecological feedback on the evolutionary processes of speciation, extinction, and dispersal between regions. So basically, it's pretty easy, at least conceptually, and now I think empirically, to explain latitudinal gradients as a function of latitudinal variation, speciation rates, and extinction rates, and then theoretically dispersal between regions, as well as we can estimate that. The influence from climate, which has several different potential mechanisms, seems, at least in amphibians, to primarily relate to feedback on community diversity. And so I think that that's one way we can try to merge these two sort of contrasting timescales of the long-term evolutionary processes that actually lead to the accumulation of the diversity, because we had to start from 1 and get to 7,000, but also the spatial variation in that diversity, which can change over very rapid timescales. I mean, if we think of, say, the glacial cycles in the last 100,000 years radically reshaped the climate in North America, the species that we have here must have been moving pretty drastically, as the glaciers came down and as the the river basins, change their course. So there have to be very short-term responses of all of these species to ecological variation. And that ecological variation somehow is feeding back on these evolutionary processes. And so with that, I'd like to thank the NSF for funding, and then GWU. And then this work was done in collaboration with my postdoctoral advisor, John Weens of University of Arizona, and Frank Burbrink at the City University of New York. And then, of course, I'd like to thank the PSW and the Cosmos Club for having us here today. And with that, I'd be happy to take any questions.
Thank you very much. I'll speak louder, we'll have a period for questions. We have 2 microphones going around. Please stand, ask the question, state your name, your affiliation, whether you're a member of the society or not, and state your question.
I have a question about latitude, climate, and geology as it would affect this all. It seems to me that you go back 50 million years, not to say 200 million years, and what was at the temperate latitudes was no longer temperate latitudes. You had a completely different geology at that time. Wouldn't you almost have to vary the geology and the climate with what you're trying to do there?
Yeah, there's a fairly large literature on the idea of effective area through time. So you're absolutely right. If we go back to, say, mid-Cretaceous, then we've got Africa's already joined up with South America, and Australia is off in the Pacific, but the Indian plate hasn't collided with the Asian plate yet. And so, and also things were a lot hotter then, as we approach the Paleocene, Eocene thermal maximum. So there's a large literature devoted to the idea that the tropics were probably more extensive in the past as well, and that as you had more effective land surface that was tropical, that probably fed back even more intensely on the generation of large numbers of lineages and species in those areas. And then what we're seeing today may actually be so even a reduced subset of that it may have been even more striking in the past, given that the tropics extended to like 50 degrees north or something. I think it would amplify all of these fundamental processes, and that's what the empirical literature seems to suggest, especially for things like angiosperms, is that on land masses that formerly had more extensive tropics, they were more species rich, related to the integration of area through time.
Hi. What did you mean by net productivity and energy? Like, what are the units? I mean...
So net primary productivity is, back here, I have it on a slide. It's measured in grams of carbon cycled per meter square per year. And that represents the net, I don't know, the ecologists will have to help me out here. The amount of carbon cycled through the environment as a function of both the solar energy received and the evapotranspiration, I think. One of the other ones is actually actual evapotranspiration, which is remotely sensed by satellites, and that has different units. I'm trying to remember. So the 2 things I have are net primary productivity, which is remotely sensed. That's NPP in grams of carbon per meter square per year. And then actual evapotranspiration, which is measured in the cycle of millimeters of water per year. And those are both measured by satellites from the amount of conductance or reflectance off of the surface. And you're right if you're suggesting that they're simply correlates of really the underlying processes that are driving the actual ecosystem energy, but they seem to be pretty strong correlates.
My name is Rudy Krutar. I'm a member of the Society, and I retired from the Naval Research Laboratory 12 years ago, where some of my colleagues were very interested in towing sensors through the water, and they called them towed arrays. So maybe, but over 40 years ago, I wrote a program which would now be called a genetic algorithm. It was a program that would create a little program and change it randomly. And if the change was better than at predicting a sequence of characters than the original was, that the change was kept, and if it was worse, it was not kept. And what surprised me was that there was a big difference in what happens if it was just as good but no better. It helped the entire algorithm much if you accepted a change that was as good but no better. And this, to me, suggested that it is not the survival of the fittest, but the non-survival of the unfit. And I was surprised when I read in Darwin's Origin of the Species that that's what he was talking about. So do you have any comments on the survival, the non-survival of the unfit?
There is a large literature on that, and there's a large literature on it as it relates to ecological interactions among species, for instance. In as much as that does seem to occur in a lot of communities, at least from a superficial standpoint. I mean, you go in and you look at these species and you're like, these can't possibly be the best species. And yet you have the massive sort of uneasy coexistence of multiple species that appear to be just good enough to survive in their environment. Another example I like to give is, I mentioned earlier that the Appalachians are kind of a hotspot for salamanders. And a lot of the salamanders are montane endemics. They only occur on a single mountaintop. And so there's some bunch in Virginia, a bunch in West Virginia, where you have these single mountains like the Cheat Mountains or the Pigeon Mountain in Georgia that have a single species on them. And yet there's a bunch of other species that are kind of these marginal habitat generalists that occur across a wide area. And so you say, well, we've got, 100 species of salamander in the Appalachians, and yet a bunch of those are these really broadly ranging generalists. If they just specialized and became better adapted to the individual mountains they were on, all of a sudden we could have as many species of salamander as there are mountain peaks in the Appalachians. And yet that doesn't seem to happen because they are able to outcompete other salamanders from invading the area, but then they never specialize and never seem to adapt further to their environment. So a lot of the community assembly that we see in amphibians might be due to something similar to that. It's not about being the best, it's just about being better than everybody else that's around you, but there's no real drive to improve beyond that.
Tim Thomas, a member of the Society. I wonder about mass extinction events and why I didn't see them in your trees. Maybe they aren't. Wouldn't mass extinctions have a huge impact if some particular area was able to avoid a mass extinction? It would suddenly be the huge
Probably
area.
so. There was a paper on that in the Proceedings of the National Academy, and they were trying to reconstruct how many lineages there were at the KT boundary, which is the only major extinction, or the only mass extinction event that amphibians would have experienced. They appear to have originated after the Permian mass extinction. And I'm trying to remember what they found. I'm pretty sure they showed a convincing increase in the rate of diversification following the KT boundary, following the extinction of the dinosaurs. So it looked like in most lineages, the rate of speciation increased after all the other competitors were wiped out. It's hard to say exactly what that did to the standing fauna because we don't really have their representatives in the current phylogenies. But just looking at it from the present back, it appears that there was a big uptick in the accumulation of species after all the competitors were wiped out during those extinctions.
I'm William Worley. For comparison, what would a map of mammalian speciation, how would that compare with amphibians? Are amphibians cold-blooded or warm-blooded?
Yes, cold-blooded.
Cold-blooded. So would warm-blooded species like mammals show a quite different pattern in terms of geographical distribution?
So this is mammals, and they show almost the exact same geographic distribution in species richness, but to my knowledge, no one done the same set of analysis to ask whether or not you actually have the same differences in the rates of speciation or extinction among those areas. So it's possible, and this is sort of responsible for the confusion, among 150 years of studying this, it's possible maybe, unlike the amphibians, the mammals just originated here, and so that's why there's more species. We don't really know because no one's done this kind of in-depth investigation for mammals. but they show the same geographic distribution of species richness.
And then it seems like warm temperatures are the highest ratio of speciation is in the warmer, the tropicals. And so could we expect with the warming climate that there would be increased speciation?
I've gotten that question a number of times, actually, and I can't see any reason why not. So I mean, what you tend to see, at least with the studies that have been done, is that when climate changes rapidly, you tend to lose a large part of your standing diversity. And some things are more sensitive to that than others. Amphibians tend to be very sensitive. So if we cranked up the heat, a lot of amphibians would immediately go extinct because they're used to cool climates. They'd be forced off the map. But just the general idea that having higher temperatures and higher rainfall leads to more speciation, it's possible that the ones that were left behind might start speciating faster. It's certainly not inconsistent with historical record. Yeah.
Would the increased energy near the tropics be caused by temperature or by sunlight? Why is there so much? Why is the tropics so energetic? Heat or light?
There you start getting into the uncaused cause sort of area of this research, which is that all of the amphibians and all the higher vertebrates and things like mammals are dependent on the existing communities that are made up of the plants and the fungi and the microbes. And so maybe if we took all of the plants away, then the amphibians would do nothing. So maybe it's actually due to the effect of the energy on the plants, on the amphibians, and then why are the plants responding to the energy? Maybe that's from the sunlight, or maybe it's from the combination of the sunlight and the water. And then it becomes turtles all the way down as we try to track it through. But that's a really salient point as to, we don't know it's specifically the sun, the solar radiation, or specifically the temperature in the environment, or specifically the rainfall. It's some correlate of that. Maybe it's actually the fungal diversity that actually promotes the insect diversity that the amphibians then feed on. But then you start getting into the questions that'll take a lifetime to answer. It's definite, but they're good questions to ask, definitely, and to try to answer. It's just we haven't gotten that far yet.
When the DNA analysis of all those species were done, were there examples of species that were misclassified?
You mean on public databases like GenBank?
No. I don't mean in that. What I mean is when they classified them based on external anatomy, it was one thing, but when you compared it by DNA, it was different. And so they said they should be in different...
I did a full-scale taxonomic revision of all amphibians, and it's made me a somewhat controversial figure in the field.
And so it was dramatic differences. Oh, very good. Okay.
Any other questions? Okay. Thank you very much. In appreciation of your presentation this evening, we have the announcement frame and signed by the members of the General Committee for you to keep.
Oh, thank you.
Thank you very much again. Thank you.